Students, good morning. Now we are going to discuss about plant tissue culture. So this is the important topic. So this is the basic of plant biotechnology. When you go for the plant biotechnology studies, the plant tissue culture technique is one of the important technique. Okay. Culture of plant cell and plant tissue in synthetic culture medium under controlled aseptic condition is known as tissue culture. Okay, do you understand this sentence? Culture of plant cell or tissue. Culture means cultivating a cell or tissue in a synthetic medium. What it is? Synthetic medium is nothing but a composition of various nutrients in which plant adapted and it grows. So that is the synthetic medium. It is prepared by uh, at a laboratory. Of course, after preparing this medium, the tissue or cell are maintained in a controlled aseptic condition. What it is? Controlled aseptic condition means the temperature is controlled, humidity is controlled, and light is controlled. Whatever the uh, physical factors available in outdoor that is controlled in the laboratory condition. We have to give artificially tube lights as a light sources and air conditioner for temperature controlling and maintenance of humidity. Okay, so culture of plant cell or tissue in synthetic medium under controlled condition is known as tissue culture. Of course, it is also called as in vitro culture. Why it is called in vitro culture? In vitro means within the vessels, glass vessels, small container. A culturing of a plant in a container is called as in vitro culture. So the culture medium provide all the essential nutrients and growth hormones necessary for growing the cell. Okay, so the controlled condition give the culture a suitable micro environment for the cell growth. Okay, so micro environment, that means controlled in laboratory. So during the tissue culture, the cells are small segment and the plant tissue undergoes repeated division to form mass of cell is called as calli. Okay. See, whenever the tissue, actually in the laboratory we used to take just 1 centimeter of tissue. That small tissue will grow as a repeated cell division as a calli, or singular it is called as callus. So callus is a mass of parenchymatous tissue which is formed as a initial tissue from the plant X plant. So each callus undergoes differentiation. Differentiation means what? It will differentiated into shoot, root, or form as a plantlet. That is called differentiation. Undifferentiation means simply remain as a cells, whereas differentiation means the tissue differentiated into shoot or root to form a plantlet. So all plantlet regenerated from plant material are identical and similar in metabolic activities. This is the most important factor as far as plant concerned. When you take the plant tissue, the tissue or cell, each cell has a totipotency nature. That means any cell grow as a mother plant. If you take a small piece of cell from root tip to shoot tip, the each cell grow as a whole plant, as like a mother plant. That is the totipotency nature. Because of this nature only, the plant tissue culture technology is evolved. Okay, so totipotency. Don't forget about totipotency of the plant cell. Whereas in animal cell it is a pluripotency. When you take the cells from a liver, it grows as a liver cell. When you take the cell from the skin grow as a skin cell, whereas plant only when you take the cell from a root or stem or leaf or flower or fruit, whatever it is, it should be plant as a whole plant, as like a mother plant. That is the totipotency in nature. Okay, all the plantlet regenerated from the plant material are identical. That means what? The plant is identical. Okay, from the single cell developed as a tissue. From the tissue you can develop more than 100 plantlet. For the example, that 100 plantlet are identical. That is the great significance, morphological and physiological. Hence it is known as a clone. So identical plant is developed, which is called as clones. So the method of raising clones is termed as micropropagation or in vitro propagation. Since we are developing a small tissue into plantlet, which is called as micropropagation. Since we are cultivating in the vessels container, it is called as in vitro propagation. So plant tissue culture has many synonyms: micropropagation, in vitro propagation, micro cultivation, and in vitro cultivation. So many things. Everything has same meaning. So plant cell and tissue are also used for production of primary and secondary metabolites. They are also used for germplasm storage. So not only plant propagation, the plant cell tissue culture technology used for production of primary and secondary metabolites. So primary metabolites what? Sugars, lipids, proteins, amino acids, and so on. What about secondary metabolites? Secondary metabolites is nothing but a medicinal compounds. That is, so many alkaloids, terpenoids, flavonoids are developed, which is called as secondary metabolites, that can be used for various applications. Can be used for coloring agent, flavoring agent, and medicine, and so on. So that can be utilized in different way. So they are also used for germplasm storage. We can develop a small plantlet or tissues that can be stored in the low temperature for future purposes. That is called germplasm storage. For the first time, tissue culture was developed by G. Abel Landit in 1902. So he is the father of plant tissue culture, who contributed many in plant tissue culture technology development. So he did not use of growth hormones in that culture medium, and whereas Fogg, F. Skoog and Co. C.O. Miller, 1975, demonstrated importance of growth hormones in plant tissue culture. So this is the important success in the plant tissue culture. So there are so many scientists contributed in plant tissue culture technology, but we are cut short with this information. Okay, so plant tissue culture is important technology where we can develop a plantlet or secondary metabolites or primary metabolites or germplasm storage. So it has various applications. So we'll discuss in detail in future classes. Thank you. If you have any doubt, you just ask now.